right, everyone. Welcome to lecture 19, where we will be discussing myogenesis, so the formation of muscles. And what you see in this really beautiful image here is actually a human embryonic hand, where in the white you can actually see the muscles forming in the embryonic hand, showing their growth. And so this is pretty amazing imagery of human development, which we don't have a ton of pictures for and live imaging for, but in this case, as technology progresses, we start to get these beautiful images, and you can see, the fingers here. And so we're going to talk about how you get this formation of this particular type of tissue. So more specifically, we're going to be focused on skeletal muscle. Here are the learning objectives covered in this particular lecture. And so we've been focused on mesoderm development this week. And we started off with the formation of the somites that derived from the periaxial mesoderm. And so we follow that down right here to the somites. And then the somites, right, are split into several different categories that ultimately result in a multitude of different tissue types. But you initially have the splitting into these three sections: the sclerotome, myotome, and dermatome, right? And we focused here on cartilage and bone that forms from the somites in lecture, I think that was at lecture 18, because it's 19. And so today we're going to be here, right, talking about the cells that start off as the periaxial mesoderm, get designated as somites, and then get further designated and specified as the myotome that'll eventually become the skeletal muscle. And so we've seen this image before, right? I used it several times, I think in the, or at least once in this lecture 18, right? But the myotome, so here we have the somite, right? So here is the neurotube, here's a notochord releasing that sonic hedgehog, right, to specify the neurons within the neurotube. And then there's all these other factors, right, that are present, differentiating the different cell types within the somite. If you look at the somite here on the left, it shows what the tissues are going to become, right? So you have the proliferating myoblasts and the primaxal muscles here. And so we're really going to be focused on those two tissue types and how they get derived, right? And so they start off as part of the myotome, the somite, get designated to become the myotome via some very key factors, which are PAX and MyoD. They get some Wnt coming from the epidermis, so coming from those ectoderm-derived tissues, so the epidermis, and so they get to also receive BMP and FGF from the lateral plate mesoderm. And so there's a lot of factors contributing to what cells are specified to become muscle. Oh, and I think I forgot about the abaxial muscles as well. We'll be talking a little bit about that. So basically all the muscle tissue in this portion of the somites, right? And so you have MYF5, PAX, and MyoD that are really necessary in order for you to get the myotome or muscle. And so the myotome of the somites only become the voluntary muscles, so those skeletal muscles. And so the muscles that make up cardiac, so cardiac muscles, the smooth muscles, are derived from a different tissue type. They don't come from the somites. So we're really just talking about the muscles that you can control, right? You decide when to move your hand if you have typical muscle development. Right? And so we're going to be right in this section today talking about this area of those cells. And so a lot of you probably have seen this image or image similar to this muscle, where you see these striations across the skeletal muscle. Then you see these cells here associated with the muscle tissue. And so mature skeletal muscle, so adult skeletal muscle, what your skeletal muscle looks like right now, is multinucleated. meaning there are multiple nuclei present in a giant cell. And the cells are striated with thick and thin filaments, and those thick and thin filaments are actually myosin and actin, which we know a lot about, right? We know how actin helps with controlling cell shapes. And so this is how your muscles are able to expand and contract. They're these giant long cells, so there doesn't have to be this paracrine signaling that occurs across all the different muscle cells to be able to expand and contract. We have evolved to have these incredibly long multinucleated cells where signaling can happen incredibly rapidly and across a large area. And so here are those nuclei, here are those nuclei here. And those striations are the thick and thin filaments. And so how do we start off in these cells that are part of the myotome, and they're essentially these round, almost epithelial-like cells, and get those striated tissue, that striated tissue that we know muscle to look like. So how do we get from here to here? And so the process that we're going to talk about, these are, and we're going to break this process down into 3 categories, right? Determination, so determining what cells are going to become muscle. Differentiation, how do they get specified and differentiated into the muscle cells that we know and love? And then how do they mature? So how do they become multinucleated giant cells? And so let's start off with the determination portion. Right? And so you have the neural tube that's formed here. Here's the nodal cord. And there are paracrine factors that are released from the neural tube and the nodal cord. So we have from the nodal cord, the MyoD, and then a whole host of other, and the PAX, the PAX factor, which is also a paracrine. So these are those myotome cells, and they're becoming determined. Right? Then these cells then receive a signal to start dividing and start multiplying and getting more and more and more cells, because you need a lot of muscle cells, right? Depending on what type of organism. If you're thinking about humans, we need a lot of muscle. If you're thinking about a horse, you're going to need a lot of muscle, right? And so you have to multiply and get your numbers up. And there's a signal that's is received to tell the cells to enter the M phase of the cell cycle. And so these growth factors are represented here in this image by these little blue dots. And then here are the dividing cells. You can see they're kind of in the middle of mitosis here. They're in metaphase. Oh, actually, I'm sorry. Well, there's separate, those chromosomes are starting to separate, so they're actually anaphase. And so they're committed at this point to becoming muscle, but they're not necessarily differentiated. So if you were to expose these cells to other factors, you could shift them into a different fate. And so they're committed to being muscle, but not necessarily differentiated. And there were a lot of experiments done to try and figure out what these factors are that are located near the myotome cells that help them become specified. And so one of the first experiments done was taking these myoblasts, so those myoblast cells that are just starting to divide but aren't necessarily yet differentiated. So what are the factors needed to do that? So you isolate a myoblast, and in the experiment, they expose these cells to a multitude of different mRNAs that they isolated from other myoblasts. They took the myoblast, isolated a bunch of mRNAs. They converted those mRNAs to cDNAs using reverse transcriptase. So if you learned about RT-PCR in 225, or maybe in genetics, and if you took molecular biology, you most likely, or microbiology, most likely have learned about reverse transcriptase. So they make that mRNA into DNA. The little C there just stands for complementary. They attach general purpose promoters to make sure that this DNA can be transcribed. And they put this DNA with general transcription factors, or general purpose, I should say, I'm sorry, promoters, into a bunch of undifferentiated random cells. So they're not myoblasts, they're just a non-muscle cell type. and they let these cells grow and basically observed what they would become. And so when they did this experiment, they realized that only some of the genes isolated from the myoblasts could actually induce a change in these general non-muscle cells to make them into the muscle cell, a muscle-like cell. So something very similar to a muscle. So from there, they determined, okay, whatever this gene must be, whatever the cDNA codes for, right, must be the factor that helps these myoblasts become determined or differentiated. And that gene was MyoD. So MyoD is a transcription factor. Here is, I think, I'm pretty sure this is a zebrafish, but don't quote me on that. And here you can see that there's MyoD expression. The blue represents MyoD here. And what I hope you notice is, right, that MyoD is not really present at all in the midline, so where the neurotube is formed, right? It's present along the flanking sides. And we know exactly what that tissue is, right? We know that that's the periaxial mesoderm, and there are those somites starting to form, right, as we move more anterior. So that MyoD, once they determined that that gene was coding for MyoD, they went to see where it was and found out exactly where it was located and part of the periaxial mesoderm of the somates. And so a BHLH transcription factor just stands for basic helix loop helix, and that just responds to the shape. Right? And so they just group all transcription factors that have this type of shape into this category. So it's not super important. But the myogenic transcription factors are MRF4, MYF5, and then myogenin. And so these are all under the category of a myogenic BHLH transcription factor. So you have MyoD, and then these 4 transcription factors that are all very similar and all necessary to get you a muscle cell. And so they also discovered that the myogenic transcription factors dimerize. So they bind with other proteins, right, in order to turn on muscle-specific gene transcription. So you're in a myoblast and you have your MyoD that's going to come in, right? Transcription factors bind to enhancers. You have your MyoD that comes in and binds. You have the ubiquitous. These are just transcription factors that you'll find in any cell. But in order to turn on these muscle-specific genes, they need to bind with MyoD. And if you were to introduce an inhibitory protein that can bind to MyoD and block MyoD from coming in to bind to its enhancer, then you can prevent muscle-specific genes from being turned on. And so you would end up, so this is what actually occurs in all other cells. This is what keeps the other cells of the somite, the ones that are going to become skeleton, so the ones that are going to become skeleton, you're going to end up having this inhibitory factor or transcription factor bind to MyoD and prevent muscle-specific genes from being turned on. So all other cells, this is what's happening. Only in myoblasts, you get MyoD coming in with the transcription factor. So cells differentiate into non-muscle cells express these myogenic inhibitors. You don't need to know the name of the inhibitors. It's not really important. All right, and so this is how you get determination, right? And so you have this determination that must occur. And so within, in order to get determination, right, you have to have that cell division. And so these are all the factors that are in play to induce these cells to start dividing. So you have all your BHLH transcription factors specific for the myotome. So you have MRF4, PAX3, MyoD, MYF5, MRF4. So you have all those 4 types of BHLH transcription factors present and activating each other. And so you have MYF5. So MYF5 expressed in a certain type of future muscle cell, PAX3 and 7. You also have MyoD expressed in certain cells here. And so all these all these factors transcription factors, right, turn on. So for example, let's start with PAX3. So you have PAX3 that's going to bind upstream of MyoD to an enhancer. PAX3 binds to MyoD's enhancer, MyoD gets turned on. MRF4 binds to the enhancers of all two of these, MyoD and MYF5. So it binds to those enhancers, turns on MyoD, turns on MYF5. And so you can follow that arrow to see how all these genes are actually feeding back and making sure that all the factors are needed in order to have cell division and start to have these cells determined. Then cell division stops at some point, right? You don't want your cells continuously dividing uncontrolled, right? We know that's cancer. And so you have to have factors present to stop all these genes from continuing to allow these myoblasts from dividing. Right? And so you end up, so you have the myogenin protein and the MRF4 present in order to stop the cell division and to help with the cells becoming more differentiated. And then you end up having muscle-specific proteins expressed. And so now you have a cell that really doesn't have any other option, right? It's going to become a muscle cell. And so, sorry about that. And so now these cells are fated to become muscle. Once you have multiplication stopping, you have cells aligning, right? They're starting to form a line, starting to get closer to the muscle shape, right? What we know muscle to become. And once they have started to align, starting to get into a similar organization as what we expect muscle cells to be, you start to get fusion and then myotube maturation. And so what essentially happens is all these individual cells, all these myoblasts that became more determined, multiplication stopped, they started to align, they then become fused. And so you end up with a bunch of cells forming one giant cell that's multinucleated. And you still have some differentiation that's occurring at that time point. Then the muscle starts to mature and you get fusion of most of the remaining myoblasts. And at this point, you no longer have these small individual cells. Right now you have these giant multinucleated cells. And so how do the multinucleated muscle cells develop? From those mononucleated myoblasts. So there's two possibilities, right? So how do you get those multinucleated muscle cells? So one possibility is that myoblast mitosis happens without cytokinesis. Right? And so this would be similar to what we see in early Drosophila cleavage, right? We know that in Drosophila cleavage, you have that syncytial blastoderm and you have all those nuclear divisions, but no new cytoplasm is made. And that's essentially what's occurring in option one here. So you have the nucleus, genetic material gets multiplied, the nucleus divides, the cell remains the same size, so on and so on, right? Until you have a multinucleated muscle fiber. The other possibility is myoblast fusion. So you have all these single nucleated cells that will then come close to each other, create attachments, signaling occurs to tell the cells to deteriorate those plasma membrane that's present between them, and then form a large multinucleated muscle fiber. And through a series of experiments, it was determined that this could not possibly be what's happening, this myoblast mitosis without cytokinesis. And so they essentially just check to see if these cells undergo the full cell cycle, right? Are they undergoing cytokinesis? And that's pretty easy to check So you could check for mitosis factors, but you could also just determine things like phospholipid formation, right? You need phospholipids to make a plasma membrane. And so they checked for all those things and determined that this is the way that it happens. You have these factors called meltrins. So meltrins are metalloproteases expressed just before fusion. So meltrins essentially are enzymes that come in and degrade that plasma membrane and other proteins that are present there between these cells. And so that's what allows these cells to eventually fuse. And if you inhibit the meltrins, you don't get fusion. So if you inhibit meltrins, you end up inhibiting fusion. And so it was discovered that these are the key factors in allowing these myoblasts to fuse with each other. And this last slide here is actually putting together everything we just talked about. It looks very complicated, but what you really need to focus on, right, is this idea of proliferation, specification, activation, proliferation, cell cycle, and then the fusion events, right? And we basically talked about most of these, right? We were more in the specification, differentiation, and then fusion here. But I thought it would be good to have a slide here. So take some time to look at that, and if you find that helpful in your studying. Okay, so we're gonna stop there, and we will discuss angiogenesis next.